Hello everyone. Today we are going to study a beautiful topic of gene expression. Right, this is the process, and there is a theory we say central dogma. And this theory says that from DNA you get RNA, and then from RNA you get protein. Right? Okay, there are exceptions also for this. If we say reverse transcription in which from DNA RNA is made, reverse transcription, that also happens in some viruses, retroviruses. For example, HIV, this is a retrovirus. So there are exceptions also for this. But in general, what we say, what is the rule? DNA to RNA to protein. So is this the only thing that we call gene expression? No. In gene expression, it's not just protein that for which the information is there in the DNA. The information is in the DNA for making different types of RNAs also. It's not just protein. And the formation of all those things from the information carried in the DNA is known as gene expression. So from the DNA you can get different types of RNAs and you get protein. Although for making protein first you need a messenger RNA and then you will get the protein. Okay. So what are the different types of RNAs? There are different types of RNAs. See, messenger RNA, then there are the transfer RNA, ribosomal RNA, long non-coding RNA, small non-coding RNA, in which there will be micro RNA, small nucleolar RNA, small interfering RNA, right? Small nuclear RNA, and many types. You don't need to know those types. See, and second thing, I told you that from DNA you get what is made. It's not from DNA it is made. From the information carried in the DNA we get RNA, right? From the RNA, from the information in the RNA we get proteins, and in that different RNAs help. If I say transfer RNA and ribosomal RNA, that are the part of cellular machinery, the machinery in the cell, where the protein is made, not RNA, where the protein is made. And there are different other types of RNAs which will help in the process of gene expression, right? You don't need to know how they will help in the gene expression because we have not yet studied what is gene expression, and we will study that topic in full detail later. Okay. So the simple things in gene expression are transcription and translation. From the DNA you get normally RNA, and then from that you get protein, but it's not from DNA you get messenger RNA. From the information in DNA, using the information in DNA, the different enzymes help. They attach different nucleotides in the proper sequence, and you get messenger RNA, right? And then reading the information in the messenger RNA, you will get a particular protein. The enzymes will help particular information, particular sequence of nucleotides in the mRNA. According to that sequence, particular amino acid will be attached, right? If I say genetic code, what I say, genetic code. So this genetic code, or we say codons, what codon is? There is information in the DNA according what that thing, gene, and that gene is a particular sequence of nucleotides, and that's reading that sequence. According to that sequence there will be production of RNA, formation of RNA that will have particular sequence of nucleotides, and that sequence of nucleotides, see here, from the DNA we get RNA, right? So from the DNA from the information in DNA we got RNA that has particular sequence of nucleotides. Suppose these three nucleotides, so three groups of nucleotides will together make up codons. Suppose this one is one codon, this one is another codon, so there are different codons and they code for particular amino acid. See, if I say one codon is there, it is having the this will code for a particular amino acid in almost all organisms. So when protein will be formed, this code will be read by someone. And that someone is transfer RNA. I told you, transfer RNA has a very important role in gene expression. This tRNA has anticodon. We will study everything in detail when we will study gene expression. So this anticodon reads the codons, and then that particular transfer RNA having anticodon according to the particular codon, that will bring amino acid according to that codon. I said one codon is there that will read for particular one amino acid, and that amino acid will be attached. Then another tRNA will come that will read another codon, and then it will bring another amino acid. Like that there will be chain of amino acids, and what will be produced? What will be produced? A polypeptide chain that will undergo processing, folding, etc. There will be folding, see, folding, and you got this particular protein. There are proteins which are made up of different amino acid chains also, different polypeptide chains also. All those things we will study later. Okay, now simple thing is that from the DNA you got RNA, from RNA you got protein, and in that process the information that was carried in the DNA, that was read, and you got codons in the mRNA, and that according to that codon you got particular sequence of amino acid in the protein. And the genetic code is universal. That is, if the same codon is there, the codon will code for same amino acid in a crocodile also, in a monkey also, in a bacteria also, in you also, right? So it indicates that all the life on earth is related. But there are exceptions to this also. For example, there are some unicellular organisms, for example some bacteria, yeast, fungi, yeast, protists, in some, right, not in all bacteria. Like in few unicellular organisms there are some exceptions that the same codon that was coding for particular amino acid in you is coding for some another amino acid in that organism. But there are exceptions. So we say genetic code is nearly universal, not fully universal. Second thing, mitochondrial DNA. That mitochondrial DNA is different from the DNA in your nucleus, and that DNA also shows some exceptions to the genetic code. Okay. Now, so the formation of DNA to mRNA, mRNA to protein, that was gene expression, but the formation of different types of RNAs from the protein was also a part of gene expression. See, from different genes you get different things, right? Okay, we will study in detail all these things later. Now let me give you an example. If I say there are different genes and they are coding for different things. If you get some infection, simply suppose you get cold and viral flu you get, then your body needs to make antibodies, and those antibodies are proteins. For them, the genes that carry information are different than the genes that carry information to make enzyme to digest your food. So different genes carry different informations. Okay, now let us talk about the eyes. There are different things the eyes can do. So the beautiful eyes, there. Okay, our eyes has lens that is made up of crystalline proteins. The lens is crystal clear, and so that the light goes properly without scattering, the refraction is well, and for that a lot of things will happen. This crystal clear lens is actually made up of different types of cells called lens fiber cells. These fiber cells, see, if you cut the longitudinal section of eye lens, you will see concentric rings, and these rings are nothing but the rings of cells that make up your eye lens, and these cells are fiber cells, and they are arranged like the rings in onion. When you cut the tree rings, don't cut tree, this was already cut tree, okay, cut onion, don't cut trees. Okay, now see, this onion that makes you cry, the structure is like the eye lens. Okay, the lens cells are very different from other cells in your body. These lens cells need to be clear. It don't need any cell organelles or nothing or the other things, right? It needs only one thing, that is crystalline protein. So it is having lot of crystalline proteins, and to make protein you need gene for crystalline protein. So when the lens cells were made, right, from the lens epithelial cells, right, there was cell division and all, right, okay, then when the cell was undergoing the process, it was undergoing different processes, right, the gene in the nucleus gives information, and what was made, lot of crystalline proteins, right, crystalline proteins. And after the crystalline proteins were made, all the organelles were degraded, nucleus was lost, everything was lost, and now the cell is only having crystalline proteins. So that this cell is crystal clear for the light to pass properly, for refraction to happen properly. And how this crystalline protein was made? Through the gene expression, from DNA to mRNA and then to protein by the process of gene expression, transcription and translation. Okay. Done. Thank you. In the next lecture we will study genome and all. Okay, go and cut onions and cry. Okay, bye bye. Thank you.